Hi. Thanks again for your patience as we figure out how to make up some lost time from Michigan snowstorms and ice storms and weather and the resulting power outages. In the presentation that you're watching right now, I'm going to finish chapter 5, which is membrane function, and then I'm actually going to record in a separate presentation chapter 6. I would have finished chapter 5 on Friday, and then I would have moved on to chapter 6, and I'm going to do all of that in these presentations, but I'm just going to record it as 2 separate presentations so that I'm not trying to move really big files around. I think it makes it a little bit easier. You should consider pausing the presentation at points to think through questions or problems that I present to you, very much like we do in class, where we will pause and you will have some time, 2 or 3 of you together, to talk over a problem that I've presented to you. You might actually consider watching this with a couple of classmates so that you can do that discussion part. But even if you're watching it alone, you can still pause it and do the thinking through. Please note that I have put numbers on the slide numbers. It's a little bit hard to see, but it's on this particular slide, it's the bottom right. There's a little gray number 1. And I've done that so that if you have questions about what I'm talking about and you want to ask those questions in class on Monday, I'll have some space at the beginning of class on Monday to ask questions. Then you can say, hey, yeah, I have a question on slide number 6. And then I can pull that slide up and you can ask your question. I'm backing up a couple of slides so that the first few things that I talk about here are things that we talked about in class on Wednesday, but I like to do that so you're not just being thrown in and trying to get your orientation. It gives you a chance to kind of get your mind where we were, where we left off on Wednesday before we start the new material. So I will go through these first few slides a little more quickly than I did the first time just because they're review for you. Okay, let's talk about active transport. So active transport involves moving a substance against its concentration gradient. So when you're trying to decide if something is active or passive transport, the key is, are we moving this thing against, or what we might say up, its concentration gradient, or are we moving it down or with its concentration gradient? And if the answer is we're moving something against or up its concentration gradient, then we're doing active transport. And the most obvious example is what we see here, what we call primary active transport. We'll talk about secondary in a moment, but primary active transport uses ATP as an energy source, directly uses ATP as an energy source. And you see that in this image. You have sodium, the sodium-potassium pump, pumping sodium from an area of low concentration to high concentration, so that's against its concentration gradient. It's also pumping potassium against its concentration gradient, area of low concentration to high concentration, and that requires an input of energy in the form of ATP. Okay, again, covered this on Wednesday, so I'm going to go through pretty quickly, but we see how this works. We see this carrier protein that is open to the intracellular fluid, the ICF, and when it's in that conformation, when it's in that shape, then it has a high affinity for sodium, and 3 sodiums are going to come in and bind to those high affinity sites. ATP, this is active transport, so ATP is going to come in and it is going to phosphorylate that carrier protein, and when it does that, it's going to cause a conformational change, so it's going to change its shape, and when it changes its shape, then those 3 sodiums, the sites where those were bound are now become low affinity. They're not as attractive to sodium anymore. So they're going to release the sodium. Sodium is going to go to the extracellular fluid. And then some sites that are high affinity for potassium are going to open up. And we're still open to the extracellular fluid at this point. So those potassiums are going to diffuse in from the extracellular fluid into the little carrier protein. They're going to bind there. That's then going to cause, again, a conformational change, shape. And we're now going to be open to the intracellular fluid, and those 2 potassiums are going to be released to the intracellular space. Okay, so then secondary active transport. With the active transport that we just saw on the primary active transport that we saw on the previous slide, we directly used ATP to pump things against their concentration gradient, which means that we set up a concentration gradient. And so there is potential energy that's going to be stored in a concentration gradient. Because when things start flowing down their concentration gradient, or with their concentration gradient, then that is a kinetic energy. So energy due to the movement of molecules. And so we can then use that energy to move other things against their concentration gradient. So we see that this is indirectly driven by ATP because ATP is used to set up the concentration gradient to begin with. But then that concentration gradient can be used, again, the potential energy in that, when it then goes to kinetic energy, can be used to move things. This can involve moving things in the same direction or in opposite directions. So meaning whatever thing, let's talk about sodium for a minute, if sodium is flowing down its concentration gradient, it might be moving something with it, or it might be moving something in the opposite direction. So if it's moving something with it, that's going to be symport. If it's in the opposite direction, then it's going to be antiport. So an example is the sodium glucose secondary active transporter, the SGLT. And in this case, sodium goes with its concentration gradient, and glucose goes against its concentration gradient. So where is ATP involved? You can pause the slide, think about that for a minute. And then I will tell you that ATP is involved in setting up the concentration gradient of sodium to begin with. Okay, practice question for you. Hold on, I'm trying to get to the right spot here in my notes. Here we go. Okay, so name two ways that active transport by the sodium-potassium pump differs from transport by the sodium-glucose secondary active transporter. So pause the presentation and think about that for a minute, come up with a little list of things, and then turn it back on. Okay, so what are some of the things that you might have come up with and how these two transport mechanisms, how they differ? Let's see, you might have said that the number of molecules moved is different. So in the sodium-potassium pump, you're moving 3 sodiums and 2 potassiums, so that's 5 molecules of something being moved. But with the sodium-glucose active transporter, it's 2 molecules at any given time, so sodium and glucose being moved. And that would be right, they do differ in that way. You might have also said that the size of the molecules being moved are different. So with sodium-potassium, you just have these 2 little ions being moved around. But with the sodium-glucose secondary active transporter, you have sodium, which is again a little ion, but then glucose, which is relatively a big molecule compared to little ions. And that would be right too. But hopefully you also thought that one of the ways that these differ is how ATP is being used. So with the sodium-potassium pump, it's being primarily used to move these molecules. But with sodium glucose, it was being used in a secondary way. It had been used to set up some concentration gradient, and now that concentration gradient was being used to do the work. Another thing you might have said is that the sodium-potassium pump is an antiporter in that it moves the molecules that it's moving in opposite directions. So it's moving the sodium to the outside of the cell, and it's moving the potassium to the inside of the cell. So that's an antiporter, whereas the sodium glucose secondary active transporter is going to be a symporter. It's moving those 2 molecules in the same direction. So the most important, or the differences that I hope that you come up with or remember or highlight, are how's ATP being used and the direction of movement of these molecules. The opposite directions are in the same direction. Okay, so now we're going to move on and talk about other modes of transport besides what we were just talking about. We were talking about osmosis and tonicity, and then we talked about diffusion and protein-mediated transport. Now we're going to talk about other modes of transport. Okay, this one we're going to skip, actually. This is in your slide set, but we're going to, for the sake of time, we're going to skip this one. Okay, now let's talk about epithelial transport. It's hard for me to say for some reason. Okay, so just to remind you, where is epithelial tissue in your body? Yes, there's the obvious example of your skin, But it's also lining surfaces in your body. It lines hollow organs, it lines blood vessels, and so that's what we're going to be talking about now. So that's pretty important there. So as we think about epithelial transport, everything we've been talking about so far, we've been talking about how do things get moved across the cell membrane. And just keep in mind that when we're talking about epithelial transport, we're talking about moving something all the way across an epithelial layer. And so there we're talking about crossing two cellular membranes. Now, we can have what we call paracellular transport. That's going to be where we're moving things between cells, not through all the way through the cells, but between them. And some types of cells allow this. We particularly see this with small capillaries and those kinds of things, but others don't. For instance, your bladder. Your bladder is lined with epithelial tissue, but those cells have these tight junctions. They're very tightly bound together, and they do not allow leakage, which is good. You really don't want urine that's being stored in your bladder to be leaking back out into your pelvic or abdominal cavity. That would not be so good. So they don't do that. There are certainly times where paracellular transport is between cells is important in your body. But most of the time, what we're going to see is transcellular transport. So that's moving through the cells. This is usually one way. We have things going one way and not the other way. And that's primarily the one we're going to be talking about. You can be aware of, but we don't need to know a lot about transcytosis. So that's where you're using vesicles to move things all the way across the cell. So let's note here, let's see if I can get this working. I'm going to turn on a little highlighter. No, maybe a pen. Is that what I'm going to do? Yeah, turn on this little pen function and see if I can make this work. Okay, so you can note here that we have what we call the apical membrane. Oh, sorry, that was terrible. But anyway, I was trying to circle that with my pen. It didn't go so well, but there it is, the apical membrane. And what that means, so let's look at this for a moment here. We see that here's some epithelial tissue. So we see these epithelial cells that are lined up together here. And we see that this says the lumen of the intestine or the kidney. So this is again, lumen being that space in a hollow tube or organ. And so here we're going to see how things end up getting across these cells. And so this side we call the apex, the apical part of the cell. And we'll see this a little more carefully in one of the next slides. But the apex, we also will call this the mucous membrane. So this will be the mucosal side or the mucosal membrane or the mucous membrane. And then we see down here, this is what we call the basolateral membrane. So it's not the one that's open to the lumen, but it's the part that's going to be open to the extracellular fluid. And believe it or not, there's actually three surfaces here. So here, right here, is going to be that tight junction, and I'm going to start drawing with this little red pen. So below that tight junction, so up here above the tight junction, that's the lumen with the intestine or the kidney or whatever it is. And so that's going to be the apical part of the cell up here. And then I'm going to put my little pen, my little red pen dot down here. And I'm now going to draw, and I'm going to go 1. So there's the surface of the cell. Here's the surface of the cell, 2, and then here's the surface of the cell, 1, 2, 3. So we see that the other 3 sides of the cell are open to the extracellular fluid. And that's going to be the basolateral membrane. You'll also hear it referred to as the serosal membrane. Okay, so talking about transcellular transport, let's look at that a little more carefully. Next slide. Having a hard time getting it to go. Come on, next slide. There we go. Okay, so now we're going to look at transepithelial transport of glucose. By the way, this would be a really good figure to use on a test because there's lots of things that we can test you on from a figure like this. So I would say that that's an important figure. So at the top of this slide, sorry, I think maybe being recorded here, some of my messages that are coming in at the moment. I'll try to ignore that. Anyway, at the top of the slide, again, we're going to be in the lumen of a kidney or intestine. And we are going to notice that these cells, so here is, I'm going to draw again red around the cell. So here is the cell like this. So we have one cell that's completely in our view, and then it's we see that it's connecting with a couple of other cells. So this is one cell here. So we see that these cells are what we call polarized, and that means that there are different properties to the different sides of this cell. So again, this part up here is going to be our apical portion. That's going to be the part that's going to be open to the lumen of the kidney or intestine. And then we have all this other side, the sides, the other three sides, that's not the apical portion, is going to be that basolateral. And essentially, when we say that they're polarized, it means that those different parts of the cell, or those different sides of the membrane, are going to have different properties. And we will find, so for instance, in this one, we see that sodium-potassium pump being on the basolateral membrane, and it usually is. We're usually going to find that on the basolateral membrane. And then we see up here on the apical part, we see that glucose-sodium, that co-transporter, right, in that part of the membrane. And so these are located in different parts of this cell, and that gives us different properties to these. This is going to allow one-wave movement of certain molecules across the membrane. And in this case, we're going to be talking about glucose. So, number one up there at that sodium glucose symporter, we see that it's going to bring glucose into the cell. So remember that glucose is going to be, well, initially in a high concentration, but it's going to start moving once glucose has just naturally flown down its concentration gradient, and then it's no longer in relatively high concentration, then we need to still have a way to get this glucose in. It's either being absorbed through your intestine, where it was broken down from your food, or we keep saying that this is the lumen of a kidney or intestine, or it's in the kidney, where remember that when the glomerulus filters things out of your blood and into that filtrate, glucose ends up going into that filtrate. Well, you do not want to lose glucose to urine. You don't want the glucose going out in your urine, so you actively reabsorb that glucose, and so that's through this process as well. So we have the sodium glucose symporter bringing glucose into the cell against its concentration gradient. So that's going to be an active transport, but it's a secondary active transport. And then we have this GLUT transporter, where it's going to then transfer the glucose to the extracellular fluid that we see down here below. Now, we need to keep in mind that this cell right here, does it have energy needs? Sure it does. It has some energy needs. But it does not need all of the glucose that is passing through it. There is going to be a lot of glucose passing through this cell, and it does not need to be using all of that for its own energy needs. And so, unlike what we saw before, where we saw that we were putting a phosphate group on the glucose, like gluten 6-phosphate, in order to keep that glucose in the cell and to keep it available there for the energy use of that cell, we're not going to be doing that. And so we get this high concentration of glucose that's building up inside the cell. So now it can just simply diffuse down its concentration gradient with this GLUT transporter. Okay, so this is not an active transport here. This is just passive going down its concentration gradient. But we call it facilitated because glucose it's not going to permeate the membrane, the phospholipid part of the membrane. It needs a protein to allow it to pass through. And so it's going to be facilitated diffusion, but it's going to be passive. And then we see the sodium-potassium pump here on the basolateral membrane, and it is the thing, of course it's using ATP, and it is the thing that's setting up the concentration gradient in order to make this glucose-sodium symport possible. Okay? All right, I think that's about all of that. Let's keep going. All right, now we're going to talk about resting membrane potential. All right, get to the right. spot here in my notes. Okay, so what is a potential anyway? Well, it's going to be a voltage difference between any 2 points. So when we talk about the resting membrane potential in a cell, it is going to be the electrical gradient that exists between the intracellular fluid and the extracellular fluid. And when we talk about this, we are always comparing the inside of the cell to the outside. So we talk about inside first relative to outside. Those are the 2 things that we're going to be comparing. Okay, so let's look at the first image here on the left. And if we were to look at this image, we would see that the cell has no membrane potential. What that means is that there's an equal distribution of the positive and negative charges inside and outside the cell across the membrane. So if we were to count them up, for every negative ion on the outside, we would have a positive. And the same for the inside. For every negative, we would have a positive. So you can count up all those little pluses on the inside, and we see 1, 2, 3, 4, 5, 6. And how many negatives do we see? 1, 2, 3, 4, 5, 6. So there we go, neutral. And same thing on the outside. However, you might notice that even though there is no electrical gradient, there is a chemical gradient. So for instance, the little purple diamonds inside the cell on the image to the left, that represents potassium ions. So notice that there are none on the outside. There are only those on the inside. And notice the sodium, that's the little circle, kind of, I don't know what color that is, orange or something, with the plus. Notice that those are all on the outside. They're none on the inside. So while this is electrically neutral between the inside and outside, we do have a chemical gradient, because sodium is not in equal proportions, right, on the inside and outside, nor is potassium. And so there is a chemical gradient. So if we were to insert, now we've moved over to the image on the right, if we were to insert a leak channel, so just a little channel into the membrane that allowed potassium to flow where it wants, then what's going to happen? Well, we have a concentration of potassium on the inside of the cell, a high concentration compared to the outside, and we have a low concentration on the outside. So what's going to happen? Well, it's going to start moving down its concentration gradient. So potassium is going to start to leak outside of the cell. Now let's think first about just that first potassium. When that first little potassium ion moves outside of the cell, what's going to happen? Well, one of the negatively charged ions that was on the inside, it's kind of like, think about its counterpart, negative, is going to want to follow it. But it can't, because notice, let me get my little pointer going here again. Notice here that we have a large anion, that's what we're saying is inside that cell. And so these are going to be big negatively charged proteins. And they can't move through that little leak channel that we've made for potassium. That's good for letting potassium through, but not those big negative proteins. And these are big negative proteins. They can't just diffuse across the cell either. So we have potassium, again, just thinking about this one for a moment. As soon as potassium left, because it was flowing down its concentration gradient, as soon as potassium left, now all of a sudden we have a difference in charge between the outside of the cell and the inside. Now we have a +1 charge because the potassium's out here. And there's now, if you counted up all of the positive and negatives on the outside of the cell, we would have one more positive than we did negative. And we have a relative negative charge on the inside of the cell. So again, if we counted these up, we would see that we have one more negative charge than we do positive charge. So now all of a sudden we have an electrical gradient. There's a difference between the electrical charge of the inside and the outside. And that's a gradient, and we can express that as saying that there's an electrical gradient, or we can also call it a potential. Those mean the same thing. So you might also note that when this happens, so another way of thinking about this this electrical gradient. So what happens with differently charged things. We know that opposites attract, right? And that the same charges like to repel each other. So notice that when this potassium leaves, and why did it leave? Because it was flowing down its chemical gradient. Then all of a sudden we can create an electrical gradient, which means there's a negative charge in here, and this potassium is positively charged. It's going to want to go back inside. So the electrical gradient is wanting to pull it back inside, but the chemical gradient is wanting to pull it out. And so make note that when something, when an ion is flowing down its chemical gradient, it's creating an electrical gradient that opposes the chemical concentration gradient. Those are wanting to pull that ion in opposite directions. By the way, we call this electrical gradient plus the chemical gradient the electrochemical gradient. And that's going to be taking into account what's happening with the concentration of this thing, as well as electrically what's happening. You should have covered this in Biology 141 and 161. So if you need to do a little bit of review on that, you can look at those notes, or you can look in your current textbook. Your Human Physiology textbook does a good job of talking about that as well. Okay, I'm not quite sure if this worked, but this slide was all crazy and the little images were covering up the text, so I stopped the recording and I adjusted the images, and so here we go. So I'm not quite sure if you're going to hear the stuff I said before or not, so I'm going to say everything again. Okay, so here we are talking about resting membrane potential. So when we talk about resting membrane potential, we often think about neurons or muscles because it's really important the way those cells function, that we know something about the resting membrane potential and how that ends up being used to do the work that they end up doing. But do know that all of the cells in your body have a potential. And in fact, all of the cells in your body do end up using that potential to communicate in an electrical way. In fact, even plants, cells will have this potential, and they end up communicating in electrical ways as well, even though they don't have a nervous system. This has been a really exciting discovery in biology. But here we're going to talk about a neuron for a minute. And so the resting membrane potential of a neuron is what we say it's negative 70 millivolts. So this is actually an average. It can range from negative 40 to negative 90, but negative 70 is the average. And why is that? Oh, and by the way, remember, so we express it as negative 70 because, hold on, let me turn on my little pen again. I wish I had a laser thing, but anyway, this is what I have. So here we see the negative 70 it's an expression of the voltage difference of the inside compared to the outside. So the inside is, relatively speaking, negative 70 millivolt difference, okay? So why is that? Well, we have negatively charged proteins. So remember, proteins are relatively big, certainly compared to ions, but these negatively charged proteins inside the cell, and then we have an unequal distribution of ions across the membrane. So we have more sodium outside the cell and more potassium inside. And again, we can thank the old sodium-potassium pump for that. And so we have an electrochemical force on sodium. Why do we say electrochemical force? Well, there's the chemical concentration difference with it being concentrated outside the cell, but sodium is a positively charged ion, right? And it's hanging out outside of the cell, and the inside of the cell is negatively charged. So we have both the chemical gradient and the electrical gradient wanting to pull it inside the cell. So we say that there is a strong force to enter the cell. For potassium, which is concentrated inside the cell, we see that the chemical gradient is wanting to cause it to leave the cell because it's more concentrated in the cell than outside. But think about the chemical component, that it's more negative inside the cell. So potassium is wanting to be inside the cell from a chemical, from an electrical standpoint, but it's wanting to leave the cell from just a chemical concentration standpoint. So we have what we call the equilibrium potential. Now you do not have to be able to calculate these, but I'm going to kind of tell you what they are. So the equilibrium potential is when the membrane potential, it's the membrane potential that exactly opposes the concentration gradient on a given ion. So again, if we think about sodium for a minute, we see that sodium is wanting to come into the cell when we just think about its chemical concentration because it's concentrated outside of the cell. But we see that it is wanting to, hold on, what are we doing here? I'm going to pause for just a minute. Okay, sorry about that, I'm back. Okay, so we see that sodium is wanting to wanting to come into the cell based on its chemical gradient because it's going to be pulled in because it's concentrated outside of the cell. But we see that also because the cell is negatively charged, that it's going to want to be pulled into the cell, right? So in that case, we see that it's pulling them in the same direction. And so what this means, this negative 60 millivolts, it means that the chemical gradient is wanting to pull sodium into the cell. Well, what would we have to do electrically in order to oppose that pull based on the chemical gradient? What would we have to do electrically? Well, we would have to have the inside of this cell be positively charged, right? Because as long as it's negatively charged, it's going to want to pull sodium into the cell, which is the same thing that its chemical concentration gradient is doing. So the way things are set up, we have them both going in. So if we were to want to set up a potential that would keep sodium basically going into the cell and out of the cell at the same rate, so where we're wanting to set up the chemical gradient so it exactly, we want to set up the electrical gradient so that it is exactly opposed to the chemical gradient. So we would need something that's wanting to push sodium out. And in order to do that, we would have to be positively charged at 60 millivolts. So we see that the equilibrium potential of sodium is 60 millivolts. Now, in thinking about potassium, what is the chemical concentration gradient trying to do? Well, remember that it is higher on it's concentrated on the inside of the cell, and so it's wanting to leave the cell, right? So what would we need to do to exactly oppose it so that it would kind of be neutral, that the force pulling it in and out would be the same? Well, if the inside of the cell was negatively charged at negative 90 millivolts, then that would be exactly its equilibrium potential, which means at what chemical potential would its concentration gradient be exactly opposed? That would be at negative 90. And so we see that potassium ends up playing a much bigger role in establishing what the actual resting membrane potential of a neuron is. So negative 70 millivolts on average is what we see inside the neuron, and we see that that is much closer to the equilibrium potential of potassium than it is of sodium. Okay? Okay, so on this slide, we can see a little bit more of what I talked about on the previous slide. So the top bullet point there says blank can leak into the cell. So what is concentrated on the outside of the cell wanting to leak in? Sodium. So sodium can leak into the cell, and potassium can leak out of the cell, but not at the same rates. So most cell membranes are 40 times more permeable to potassium than to sodium. And potassium flowing out of the cell causes the membrane potential to be negative 90 millivolts. So remember that potassium is trying to flow out because it's concentrated inside the cell. So at what point does the electrical gradient that it is creating, that's wanting to pull it back inside the cell, at what point do those become in equilibrium and you have equal numbers of potassium leaving and entering the cell? Well, that's at negative 90 millivolts. But that's not the only ion moving around here that we need to consider when we're thinking about what the resting potential of this membrane is going to be. Sodium is also going to be flowing into the cell, but remember, it's the membrane is not so leaky to sodium as much as it is potassium. So it's a much weaker force, but it is going to offset that negative 90 millivolts a little bit. So we end up with the resting membrane potential of a neuron at negative 70 millivolts. And then, of course, we have the active transport of sodium and potassium using the sodium-potassium pump. And that's also going to help maintain this negative membrane potential. Instead of just thinking of it as 3 sodiums out and 2 potassiums in, think about it as 3 positively charged ions out and then 2 positively charged ions in, so that you can see that, that the net there is essentially going to be one more positive on the outside than on the inside, right? So that's helping to maintain the negative membrane potential. Okay, on the next slide is a little video, which I think is helpful in thinking about the resting membrane potential. And I think it's going to work for me to show the video right here in this presentation and for you to be able to watch it. If it doesn't for whatever reason, I do also have it available on the Moodle page, so you could just watch the little video separately, but I hope that it's going to work right here in the presentation. So let's see if it does.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, that's it for this presentation, and we will See you at the next one.